So, hi there, friends of the Italian opera. I decided to stop singing now. Some colleagues told me I can play, but stop singing. So I followed them. Also, there are people like Donald Trump, everyone knows that he's an awful president, but he was doing it for 4 years and he still has 70 million followers. Even he was the most awful president, so I'm probably the most awful singer. So why shouldn't I sing a song? Anyhow, let's continue with science now. Cancer stem cells is the topic today, so it's the third part of the lecture for you master students in radiation biology. So the second part was finished with the typical markers for cancer stem cells, like CD surface markers, you may remember, can be easily detected with antibodies and some overexpressed transcription factors like SOX2 or OX34. They are usually detected with either RNA sequencing or with some reporter genes, and there were also some functional markers. Okay, so the third part will now focus more on the relevance for cancer stem cells or of cancer stem cells for the problem of tumor therapy for oncological practice. And the most severe problem in oncology is, of course, the development of tumor cell resistance, so of some tumor cells that escape therapy, could be chemotherapy, immunotherapy, radiotherapy, of course, and which survive all the therapeutic interventions and form a second or a residual tumor later on. So this means that even if you are able to kill 99.999% of all the tumor cells, if there's a single surviving tumor cell, and this surviving tumor cell has cancer stem cell potential, it with high probability leads to a residual tumor later on. So what are the different targets of tumor therapy, chemotherapy, radiotherapy, immunotherapy? So you can, of course, we know that the tumor is heterogeneous. It contains different kinds of cells. Among them are these cancer stem cells, and there are the bulk cancer cells surrounding them in niche structures or just in other structures. So what conventional tumor therapies are doing, they target the majority, so the bulk tumor cells. Yeah. So chemotherapy, radiotherapy, their oncologists are happy if the tumor starts to shrink, and it shrinks to such a degree that after completion of the therapeutic procedure, there is virtually no tumor detectable anymore. But no tumor detectable doesn't mean that there are some surviving cancer stem cells, which is shown here on this lower part. So if there's a single cancer stem cell surviving this tumor therapy, it with high probability will later grow back to form residual tumor later on, and this of course means therapy failure. The hope for the future, of course, is that we will have drugs or other therapeutic measures, including radiotherapy, that at least, among others, can also kill the tumor stem cells. Yeah. So killing of the tumor stem cells in the future is the biggest goal that we have to achieve in oncology, because if we are able to kill the tumor stem cell, then finally, after a certain time, the tumor will shrink and completely degenerate. Yeah. At the moment, most therapeutic measures, they use this path. So they try to quickly shrink the tumor mass and are not really able to target these cancer stem cells. So why are cancer stem cells sometimes more resistant than the bulk of the tumor cells? There are some intrinsic and some extrinsic factors. The intrinsic factors means factors that are autonomous to the cancer stem cells. So I already mentioned before that they are usually characterized by high expression of these efflux pumps, ABC transporter, so C stands for cassette, by the way. ABC transporter means ATP binding cassette transporter. Yeah, so there are about 6 members of the ABC transporter genes that code for efflux pumps on the membrane. MDR is one. It was the first was discovered and was named multiple drug resistance because if this is highly expressed on the membrane of tumor cells, it pumps so many of the chemotherapeutics out of the cytoplasm that the cancer cells become really highly chemoresistant. Cancer stem cells also express other detoxifying enzymes. Aldehyde dehydrogenase, I already mentioned. Glutathione S transferase is another one, and cytochrome P450. These are all detoxifying enzymes. Usually, outside of the tumor, they are highly expressed in liver because liver cells always have to detoxify many xenobiotic compounds. But expression in the tumor makes the tumor cells, tumor stem cells, highly chemo resistant. I also mentioned already DNA repair, so that um, cancer stem cells have a high capacity to quickly repair DNA damages without taking too much care of the fidelity of the DNA repair. So there it tolerates mutations, it tolerates small deletion, doesn't matter, but at least to a degree that makes the cell surviving, makes the cell able to divide. Uh, DNA repair is um, uh, highly expressed in cancer stem cells. But there are also extrinsic factors, so factors that are not really located inside the cancer stem cell, but outside, and this is its association within a protective stem cell niche. I already mentioned this. So this stem cell niche provides a low oxygen environment, and this low oxygen environment is instrumental to protect the cancer stem cells from the influence of um, cytotoxic oxygen species. So I will focus the rest of my talk mainly to the problem of the high expression of the efflux pumps or the ATP binding cassette genes. Um, Multiple drug resistant gene is one of them. Um, Others are called ABC G1 to ABC C6, I think. So as I said, these genes, they code for proteins which reside on the cytoplasm, on the cell membrane, and driven by ATP as an energy source, these efflux pumps pump out xenobiotic molecules. And among these xenobiotic molecules are in particular uh, cytostatica, such as doxorubicin or adriamycin. So they are preferentially pumped out. So if they usually passively diffuse in the cell and are of course intended during therapy to kill the cell, but the cell has this self-protecting mechanism, this pump, which continuously pumps them out. This high expression of the ABC efflux pumps we can use as a marker, as a functional marker, I already mentioned this, to identify cancer stem cells. And how this works is shown here. So we use the method flow cytometry, particularly with a sorting application. First, just by staining the tumor cells, we use a dye. It's called Höchst. It's a DNA major groove binder, so it's a DNA dye. Höchst is 342, and this Höchst has virtually two. If it's excited with UV light in the flow cytometer on the FAX sorter, it has two emission wavelengths. One is more blue, and the other is more reddish. And if you have the Höchst bound to the DNA, that is what we usually use it for, then you have a virtually balanced red and blue fluorescence from the Höchst dye. If the Höchst dye is still outside of the nucleus and freely in the cytoplasm, it has a relatively stronger red, so it appears more purple. So you see it here in a flow cytometer. So when you analyze tens or hundreds of thousands of cells, each represented by such a spot here. So unfortunately, the cursor now not works again. So you have here cells that make up a so-called site population. Yeah, so these are few cells here. There's about 4% of all the tumor cells that were analyzed here from 100% or 96% of all the cells are here, binding Höchst to the nuclear membrane with the equal red and blue fluorescence. But then you have this site population. For some reason, some cells have a relatively reduced blue fluorescence uh, with an unchanged red fluorescence. And this is these are cells that um, have a reduced concentration of this Höchst dye in the cytoplasm. And this reduced Höchst concentration in cytoplasm is because these cells have a high expression of these ATP pumps. Yeah, and we suspect that the high expression of the ATP pumps is specific for tumor stem cells. We can assume that the site population seen here in flow cytometer with Höchst on the FACS sorter represents the cancer stem cells. And this can be also tested if this has really something to do with the expression of the ATP binding cassette efflux pumps by adding a inhibitor. This is verapamil. It's a drug used in cardiology, and this is an ATP binding cassette inhibitor. So if we add this inhibitor here on the graph on the right hand side, we see that the site population cells disappear. Yeah, so they no longer can pump out the Höchst dye out of the cytoplasm. Yeah. So the nice thing here is that this works virtually with live cells. So this Höchst dye is not toxic. Yeah. Also the cell tries to pump it out, but it is actually not toxic. So we can use those cells after sorting them with a flow cytometer FACS sorter and do further experiments. So this population, it's called here R4, this is a site population. We have sorted out, and let's see what we did now with them. So we sorted out those cells, living cells, run through the flow cytometer, through FACS sorter, and took these site population cells where we suspect these are cancer stem cells, put them again in culture, and you see they're nicely growing. So the red color here, it has nothing to do with the Höchst dye. This is another, it's a fluorescent protein that we labeled the cells um, um, permanently with. And you see one interesting thing, these cells on the site population, they grow in a particular pattern and they form these spheroids. And because the cell line that we used here originally was a bone tumor cell line, osteosarcoma cell line, we call these spheroids sarcosphere. Yeah, they come from sarcoma and are spheres, grow in sphere-like fashion. So they are sarcospheres. And sarcosphere are an indicator for cancer stem cells from sarcoma. Yeah? If you sort out from the flow cytometer the cells that constitute this main population, so with a high expression of Höchst or with a high concentration of Höchst in the cytoplasm, you then see that growing them on plastic, the cells are more flat, so they are the more conventional cells that you usually see on cell culture with the tumor cell line. So when we then put these cells into a xenotransplanted mouse, yeah, so this is a congenic mouse here, just shaved, removed of the fur, these cells here from the site population injected into the back under the skin subcutaneously into the mouse. Then after 10 days, so the mouse is here visualized after 10 days with a fluorescent camera, you see a huge tumor growth here under the skin. When we inject Same I think was 1 million cells here from the main population, yeah, from the more differentiated tumor cells. Actually, nothing happened. So the main population cells are not really tumorigenic anymore. So this shows that indeed the site population cells are propagating the uh, tumor growth. Yeah, so they are initiating the tumor growth in a transplantation model. We can do similar experiments here in vivo, and now we take the site population cells from two cell lines virtually. We have one cell line where the cells are fluorescently labeled with fluorescent protein in red, and another, oh no, this one, are labeled in red with a fluorescence protein. So cells are permanently glowing in the dark under the microscope nicely. And we have another cell line where the cells are not labeled fluorescent. Then we do the Hoechst staining in both, FACS sorting. So from one cell line we took the main population. Yeah, the main population is unlabeled. The site population from another cell line was fluorescently labeled in red, and we mix them in the ratio as they appear here in the normal conditions and plate them out. And what we see after 3 days, okay, we have a few of the site population cells because they are lower in number, of course. Yeah, in the past we said about 4%. It's maybe 4% here. Not so frequently. You see, mainly we have main population cells, so that means non-cancer stem cells. However, if we wait long enough, and we waited here 14 days, passage several times, then suddenly what we see here is that after 14 days, the cells that were originally a vast minority, just 4%, yeah, site population cells here in red, after 14 days, they start to overgrow everything else. So they, on the long term, they really have a growth advantage, yeah. So the main population cells, also they were larger in number initially, they virtually become less and less over time. So this just shows that on the long term, the site population cells fulfill the criteria of cancer stem cells in the sense that they are the cells with a long-term repopulating potential. But they are also important for chemotherapy, for the success of chemotherapy. And this is shown here with this study on neuroblastomas, or a tumor of the brain of the malignant neurons. They are notorious for very frequently developing chemotherapy resistance. And the study here involved two cell lines from neuroblastoma. So one was a good responder, a cell line called JF, good therapy responder to chemotherapy. And the other one, IMR32, was a poor therapy responder. And to the people who did this, they also used the site population measurement with flow cytometry. Um, and so what they did, they from both cell lines, they um, purified, or they sorted the site population cell and the main or non-site population cell, and checked them for the expression of these efflux pumps. Yeah? And you see it here. Yeah, you have the good responding JF cell line. Here the site population cell. Here the main population cell, and here always the expression. Yeah, so this is delta delta CT. It's a parameter for the gene expression level. Yeah, and you have selected here 3 particular genes that code for these efflux pumps. Yeah, MDR1, or I mentioned before, ABCG2 and ABCG1. And you see in the good responding, so in the relative sensitive neuroblastoma cell line, the genes, these 3 genes, are slightly overexpressed in the site population. Um, so this is one indication, but relative to the resistant cell line, the expression level is not so high. If you look in the resistant cell line now, so the cell line that where the cells did not respond to chemotherapy, you see that one of these efflux pumps, ABCG2, is extremely high expressed, but this high expression is only present in the cells of the site population, so of the cells that we believe are cancer stem cells. So this is probably the high expression of ABCG2 in the tumor cell line, in the cancer stem cells, is probably the reason for this tumor cell line being so chemosensitive, chemoresistant, sorry. So now they already tried to validate this hypothesis by a forward experiment. So they sorted the cell with a fluorescence assisted cell sorting technique, took they took the resistant, the sensitive cell line, I think, the JF1, the good therapy responder, and sorted site population cell again from non-site population cell, but now they used these living cells and performed a new cell experiment. So they put them in culture, treated them with a chemotherapeutic, that is mitoxantrone. This is established chemotherapeutic cytostatica for neuroblastoma, and looked how these different um, parts or these different populations of these neuroblastoma cell line respond. And what you see, the clonogenic survival colonies per well without mitoxantrone was very comparable in this main and in the site population. Yeah, here black is the main population, white is the site population. So without mitoxantrone, no big difference. But if you add the mitoxantrone now, you see that the main population cells, they are completely killed, so there's virtually zero survival, whereas the cells of the site population, uh, they are relatively resistant, so they survive uh, at 1 nanogram per milliliter mitoxantrone almost as good as the control cells. Yeah, so this suggests that the site population cells, but not the main population cells, are highly resistant to chemotherapeutic treatment. So this um, let's hypothesize now or plan some use or some search for novel drugs that might be able to target the cancer stem cells, which are the reason for chemoresistance. So one possibility that people already tried was to simply block or inhibit these efflux pumps, these ATP binding cassette efflux pumps. And I told you there is a inhibitor available that's used in cardiology, verapamil. This worked very well in vitro. Yeah, so if you use the site population cells and the main population cells and added verapamil to the site population cells plus a chemotherapeutic, the site population cells could be killed. Yeah, because the efflux pumps didn't work anymore with verapamil, and then adding the cytostatica, the chemotherapeutic drug, then killed the cells. This worked. However, unfortunately, the concentration or the dose of verapamil that had been used was so high that use in real patient or even in animal models killed the entire animal before the tumor cells became resistant, became sensitive. Yeah, so verapamil in low dose, I told you it's used in cardiology to treat, I think, arrhythmia or something in low concentration. In higher concentration, it is highly cardiotoxic. So if the concentration was high enough to make the tumor stem cells chemosensitive, the patients or the animals, the experimental animals, died from heart failure. So unfortunately, this currently this idea of specifically targeting the ABC efflux pumps um, is an interesting concept, but practically it has not worked till now. Another approach was to use so-called small molecules, cytotoxic small molecules. There's a whole there are big libraries of small molecules that are synthesized. Small molecules, in contrast to the established cytostatica, as the name suggests, they are much smaller. Because they are so small, they are usually not transported by the efflux pumps. Yeah, so once they passively diffuse into the cytoplasm, they stay there. Yeah, the cell has very little measures of getting rid of small molecules. Of course, there are big efforts to screen all these small molecule libraries for a tumor cell specific cytotoxicity. There are many found that are cytotoxic indeed, but unfortunately, they are not so specific. So when they are uh, killing tumor cells, usually they also kill normal cells. And of course, then the balance between tumor-specific cell killing and cytotoxic side effects on normal tissue would be too small. So there would be too much side effects on the patient's well-being. So this didn't work. Um, okay, then I mentioned that cancer stem cells, or I have not mentioned, but maybe you know this, the cancer stem cells, they are slowly dividing, so there are always only a few cells in S phase. Most of the time, cancer stem cells are in a resting or in a quiescent state in G0, and these cells in G0, they are particularly naturally resistant to any DNA or genotoxic agents like etoposide or 5-fluorouracil. So one idea was to somehow challenge or induce all the cancer stem cells to enter S phase synchronously. Um, and if you then give the conventional chemotherapy drugs like etoposide or 5-fluorouracil, you would also target the cancer stem cells because they were hopefully just all in the S phase cell. I don't know how far this went. Um, I probably have some progress in this. The last possibility aims to target um, the Achilles heels of cancer stem cells, and this is their high DNA repair capacity. So the idea here is to use small interfering RNAs to downregulate the high DNA repair capacity or the genes that are involved, and also use so-called PARP inhibitors. So PARP is a particular protein that is involved in alternative non-homologous end joining. I think that is frequently used by cancer stem cells. Okay, so I show you here a summary a little bit, read through. Um, Also, there was an intermediate summary before about the importance of cancer stem cells for metastasis. I mean, go through and we can discuss the topics that are given here in the summaries. Also try to remember some of the uh, information here, maybe for the exam that you will have, I think, in January, February next year. Okay, so thanks for your attention, and sorry for the bad music.